The respiratory system that is consists of number of organs. Basically, it consists of a lungs. This is a pair of lungs that we can see, and lungs is actually connected by a tube. That tube is known by the name trachea, and on the upper side of the trachea we are seeing the pharynx, or the upper portion of the pharynx is slightly enlarged also, and that is known by the name larynx. So larynx is the part that is opening to the mouth. So when we are taking air, the air will be reaching this part. From here it is going down by this windpipe, otherwise known by the name trachea. And trachea will be dividing into two branches, and that is known by the name bronchus, plural bronchi. And we can also see that bronchi is again dividing into finer branches, and the smaller branches of bronchus is known by the name bronchioles. Now, if you take each bronchiole, it is again dividing or branching out, and finally each of the bronchiole is opening into a small sac, and this sac is known by the name alveolus. So here you can see that this is the alveolus, or the sac-like structure known by the name alveolus. And basically the exchange of air between our blood and the air is actually taking place in this part of the lungs. So this part of the lungs is known by the name alveolus or alveolar sac. It is in the alveolus basically the exchange of oxygen and carbon dioxide is taking place in our lungs. So this is an overall idea regarding the structure of our lungs, and I think you have studied about this structure from your high school classes and also in your plus 2 classes. You have some idea how the lungs is working and what is the basic arrangement or basic function of the lungs. Now today what we have to see in detail is how the transport of oxygen as well as the transport of carbon dioxide is going on. So to begin with, we will start with the transport of oxygen. Regarding the transport of oxygen, here you can see that the exchange of oxygen and carbon dioxide takes place in between lungs and the blood, and I have already shown that this is basically taking place in this alveolus. So it is in the alveolus this transport of oxygen and carbon dioxide is taking place. And what is important is that 97% of the oxygen is actually carried out by the RBC. So RBC is the basic function of the blood is to carry the oxygen, and for carrying the oxygen it has a pigment known by the name hemoglobin. Hemoglobin is the most important pigment that is helping our body or our blood to transport oxygen. So 97% is actually transported by hemoglobin or by RBC. So normally what is happening is that hemoglobin is a respiratory pigment. When it is combining with oxygen, this hemoglobin becomes rich in oxygen. When it is rich in oxygen, the color of the hemoglobin, which is normally purple in color, will change into bright red. So one molecule of hemoglobin can carry up to 4 oxygen atoms. So when all the 4 oxygen atoms are occupied by the respiratory pigment called hemoglobin, its color will be changing and it becomes bright red. And this hemoglobin is actually attached with the RBC. So it is within the RBC this hemoglobin is present. When RBC is moving around in the blood, this hemoglobin will be also carrying the oxygen to different parts of the body. Here you can see that how this all structure, all process is actually taking place. So I already told you this is the alveoli. Here in the earlier picture also I have, this is the alveoli. So if you take one alveolus, the arrangement will be almost like a balloon, and very closely connected or very closely attached with the alveolus is our blood vessel. This is the blood capillary system. So when air is reaching the alveolus, there will be air inside. In that air, there will be both carbon dioxide oxygen and nitrogen, etc. Oxygen part will be moving into this blood vessel by what is called diffusion. By through diffusion, the oxygen will be entering into the blood, and it will be absorbed by the hemoglobin of the RBC. So here you can see that the oxygen enters into RBC. Here this is also, you can see that oxygen is entering into the RBC. In the RBC, it is combining with hemoglobin. Here you can see that this is the oxygen that is entering into the hemoglobin. Here it is combining with hemoglobin. As a result of that combination, we are getting a product known by the name oxyhemoglobin. This is HbO2, which is known as the oxyhemoglobin. And the color of the oxyhemoglobin, as I told you earlier, is bright red. Normally it is purple in color. Now, so this is the method by which 97% of hemoglobin is actually transported. Here also you can find another. So normally the amount of oxygen, it is usually represented as partial pressure of oxygen. In a deoxygenated blood you might be knowing as the impure blood, will be containing around 40 mm of mercury. So that is a normal amount of blood oxygen that is present in a deoxygenated blood. When this blood is absorbing a lot of oxygen, the amount of oxygen in the blood will be increasing, and it becomes 100 mm of mercury. So from 40 there will be an increase of 60. So that is why the color is actually changing from purple color to bright red color. So this is how the process is going. So this is the method by which 97% of hemoglobin is actually transported. Now there is also another point. There is 3% of oxygen. That 3% of oxygen is dissolved in the plasma, and it is transported in that form. That is 97% is transported as oxyhemoglobin. 3% will be entering into the plasma. Plasma, you know what is plasma. And in the plasma, it will be carried in that form as a dissolved oxygen. That is the process by which the other remaining 3% is actually transported. So a small portion of oxygen also dissolves in the plasma and is carried in the form of a solution to the tissues and blood stream. So this is the method. So basically we are finding two methods. One is in the form of hemoglobin, by which 97% of oxygen is transported, whereas a small portion is only transported in the form as dissolved in the plasma. So now this free oxygen, before entering into the tissue, first passes into the tissue. So this is how the process is actually taking place. Then you can see that oxygen transport, and I have also shown here that here also we have explained that this is the partial pressure of oxygen in a deoxygenated blood. When the blood is absorbing oxygen, it is getting enriched with oxygen, and the color will be changing to bright red. That is what is mentioned here. So from oxygen transport from lungs to tissue is achieved because hemoglobin has the highest affinity for oxygen at 100 mm of oxygen, which is almost present in the alveoli, and low affinity for oxygen at 40 mm of oxygen. So this is the situation that is present in the lungs as well as in the tissue. Then what is here also we have find another picture. Now that is indicating the partial pressure and the percentage of oxygen saturation. Now what you have to understand is that the hemoglobin is actually taken, this combination of hemoglobin with the blood is actually taking place in the lungs. Now when this blood is reaching a tissue, in the tissue an opposite effect is actually taking place. Here oxygen combines with hemoglobin, forming oxyhemoglobin. Now when this oxyhemoglobin is reaching a cell or a tissue, what happens is that there the partial pressure is only PO 40 mm of mercury. Under a low partial pressure of oxygen, what happens is that this hemoglobin will be splitting, and it will be splitting into oxygen and hemoglobin. So in the cells or in the tissue, a reverse process of splitting of hemoglobin is taking place. Whereas in the lungs, this what is happening is hemoglobin is combining with oxygen. In order for this combination to take place, the partial pressure should be 100 mm. But in the case of lungs, the partial pressure is always 100 mm. But in the case of tissue, the partial pressure will be always 40 mm. At that partial pressure, the reverse process, that is splitting of oxyhemoglobin into hemoglobin and oxygen will be taking place. And as a result, that this oxygen is released by the oxyhemoglobin and that will be absorbed by the cells or tissue. So that is what is mentioned here. So that in the case of cells, the partial pressure will be 40 mm. In case of lungs, it will be 100 mm over. So this is how the whole process is actually taking place. Now there are number of factors which are influencing this partial pressure, and that is what is mentioned here. Here you can see that there are one is carbon dioxide, 2,3-diphosphoglycerate temperature. Any increase in temperature will be actually promoting this combination, the graph to move in this direction. We will gradually see in this line it is showing the percentage of oxygen hemoglobin saturation. This is the partial pressure in mm. That is, if you closely observe that when partial pressure is increasing, the percentage of oxygen saturation, hemoglobin's oxygen saturation is also increasing. So here you can see that at 40 this was the partial, this was the oxygen saturation. At 60 again the amount of hemoglobin saturation has actually increased. Then if you look at again that at 80 this is the level, almost 90%. At 100% there is 100% hemoglobin saturation. Now there are a number of factors. One is temperature. If there is a rise in temperature from, for example, 20 to 30 degree, what happens is that this graph will be always moving in this direction. That is what is shown here. So a increase in temperature, similarly a presence of one chemical called diphosphoglycerate. More amount of diphosphoglycerate can also push this graph in this direction. That is, the percentage saturation of hemoglobin can increase. Another factor is carbon dioxide. More carbon dioxide in the blood means more oxyhemoglobin saturation. Whereas there is one factor, there is pH. If pH is increasing or a decreasing pH or pH is decreasing, what happens is that it is also pushing the graph in this direction. Now what we are seeing in this case is that hemoglobin, that is, if the anything that decreases carbon dioxide or decrease the temperature will be actually pushing this graph in this direction. That means that the percentage of saturation will be decreasing. So what is we have to understand from this picture is that if any of this factor is increasing in the blood, the percentage of hemoglobin saturation will be increasing. Any decrease in this factor will be decreasing the percentage of saturation. So that is an idea regarding this graph, and this graph is known by the name oxyhemoglobin dissociation, oxygen dissociation curve. Now, so this is an idea regarding how transport of oxygen is actually taking place. So remember that basically there are two methods. One method is in the form of hemoglobin. Another method is in the form of dissolved plasma, whereas only a small portion is taking place. And in the oxyhemoglobin, you have to keep in your mind how this whole process is actually going on. Now, another type of transport is the case of transport of carbon dioxide. So, in case of transport of carbon dioxide, it is slightly more complicated or slightly more elaborate. That is, 3 different methods are there. So, basically, the blood transport carbon dioxide in 3 methods. One has carbonic acid, other has bicarbonates of sodium and potassium, and third as carbaminohemoglobin. We will see how these 3 methods are operating. The first is that about 10% of the total carbon dioxide is carried by the blood in the dissolved state as carbonic acid. That is 10% out of the total carbon dioxide. Some amount of carbon dioxide will be combining with water in the blood, forming an acid known as carbonic acid. So 10% of the carbon dioxide will be dissolved in the water and will be carried in the form of carbonic acid. So that is one method of that is the first method as carbonic acid. Only 10% are actually transported. Now second is the more advanced method or more prominent method. Here 80% of the carbon dioxide is transported as sodium bicarbonate in the plasma or as potassium bicarbonate in the plasma, or as potassium bicarbonate in the case of RBC. So there are three methods. Is that 80% of the carbon dioxide will be transported as bicarbonate ions, either as sodium bicarbonate or as potassium bicarbonate. It can be transported either in the blood plasma or within the RBC. So that is why they have mentioned here 80% as sodium bicarbonate in the plasma and potassium bicarbonate in the plasma, or potassium bicarbonate within the RBC or within the red blood corpuscles. So that is the second method is bicarbonates of sodium and potassium. Now one more method is actually remaining, that is the carbaminohemoglobin. Now the last 10% is transported as carbaminohemoglobin. Actually carbaminohemoglobin is a very loose compound formed by the combination of carbon dioxide and hemoglobin. So when oxygen and hemoglobin are combining together, we are getting oxyhemoglobin. Similarly, when carbon dioxide is combining with the hemoglobin, it is forming carbaminohemoglobin. That is a product that is found when carbon dioxide and hemoglobin. So that is the method by which the last 10% is transported. So here you can see that there are three principal methods by which we are transporting transporting carbon dioxide. The most important method is the as bicarbonate ion, and other two are small minor method. Only 10% is transported and dissolved carbonic acid. Another 10% is transported as carbaminohemoglobin. And we will see some more details about this process, how it is all taking place. So the majority of the carbon dioxide molecule, that is our carbonate, are carried as a bicarbonate or bicarbonate buffer system. And in this system, carbon dioxide is diffusing into the blood, RBC. And here you can see that. So this is how the process is going on. See, this is the carbon dioxide. Carbon dioxide is from the tissue that it is entering into the this carbon dioxide is entering into RBC. In the case of RBC, carbon dioxide and water are combining, and for that an enzyme called carbonic anhydrase. Here you can see that carbonic anhydrase within the RBC quickly convert carbon dioxide into carbonic acid. Here you can see that. So carbon dioxide and water will be combining with each other, forming carbonic acid. Now, and for that there is an enzyme called carbonic anhydrase is required. Now what happens to this carbonic acid? This carbonic acid will be splitting into two ions. One is a bicarbonate ion, another is a hydrogen ion. So here you can see that this is a bicarbonate ion and this is the hydrogen ion. Here and this bicarbonate ion, so the is an unstable molecule. It will be splitting into bicarbonate ion and hydrogen ion. Now what happens is that this bicarbonate ion will be pushed out of the RBC. Here you can see that this is the negatively charged bicarbonate ion which are pumped out of the RBC. Now there is a remaining hydrogen ion. So this hydrogen ion is a positively charged, that will be combining with hemoglobin, forming, and there are a different type of another type of hemoglobin is simply hemoglobin also, that will be combining with another molecule of hemoglobin, and here a different type of hemoglobin will be formed. Now what happened? There is a problem here. Is that the problem is that when more amount of this negative this C, you take this bicarbonate ion. When 10 bicarbonate ions are actually pumped out of the RBC, a deficiency of 10 negative charge will be occurring within the RBC. So here this is one is going out. We imagine that 10 bicarbonate ions are moving out. So when 10 bicarbonate ions are moving out, there is a deficiency of 10 negative charge, or here the negative charge is increasing, and that will be creating some electrical imbalance. To avoid that problem, what our body is doing, that here you can find negatively charged chloride ions. When 10 negatively charged bicarbonate ions are pumped out from the RBC, 10 chloride ions will be pumped into the RBC. So here you can see that this pumping of chloride ions into the RBC is known by the name chloride shift. This is known as a chloride shift. There is another term called hamburger phenomenon. So the term chloride shift or hamburger phenomenon simply means that when a 10 bicarbonate ions are pumped out of the RBC, to maintain that electrical charge or electrical neutrality, the blood will be pumping another 10 negatively charged chloride ions into the RBC. So maintain the electrical charge equal. And that process is known by the name chloride shift, otherwise known by the name. Here the whole process is again explained in a much more detail. See, this is the carbon dioxide combining with the water, forming carbonic anhydrase. This is the carbonic acid. Carbonic acid is splitting into two products. One is bicarbonate ion, other is a hydrogen. Bicarbonate ion is diffusing out into the plasma. When 10 by 1 bicarbonate ion is diffusing out, 1 chloride ion will be diffusing in. So that is to maintain the electrical charge. Otherwise, there is a increase in negative charge within the RBC. So what happens with the hemoglobin is that it will be combining with another oxyhemoglobin, converting it into normal hemoglobin and an oxygen molecule. So the hydrogen ion will be combining with the oxyhemoglobin, converting it into normal hemoglobin and an oxygen molecule, and this oxygen molecule will be diffusing out into the plasma. So this picture you must keep in your mind. It's a very important diagram. If you know this diagram, you can easily explain how this whole process is actually taking place. It's a very important diagram. So keep diagram in your mind, how this transport of carbon dioxide is actually taking place in your blood. So this is what we have been explained. This is called chloride shift, and there is another term for chloride shift is known by the name hamburger phenomenon. So these are the idea regarding the functioning of hemoglobin, and in the case of transport of oxygen, as well as in case of transport of carbon dioxide also. So these are the three different methods by which the carbon dioxide can be transported in your blood.